Imagine being told you have terminal cancer. Surgery failed, radiation failed, chemotherapy failed. Even the newest immunotherapies couldn't stop the tumors spreading through your body. Then, doctors offer you one last experimental option. They want to reprogram your own immune cells with genetic scissors. 2 years later, you're cancer-free. This isn't science fiction. It just happened at the University of Minnesota, and the results are shaking up everything we thought we knew about treating incurable cancers. Here's what went down. Researchers took immune cells already inside patients' tumors, cells that were trying to fight the cancer but losing the battle. They used CRISPR gene editing to permanently delete a single gene that was holding these cells back. Think of it like removing the parking brake from a race car. Once that genetic brake was gone, these supercharged immune cells went to work. The gene they deleted is called CISH. Never heard of it? Neither had most people until this trial. CISH acts like an internal off switch that prevents your immune cells from going full throttle against cancer. The problem is, you can't block it with traditional drugs because it works inside the cell. That's where CRISPR comes in. It can reach inside and permanently delete the problem. 12 patients signed up for this trial. All of them had advanced gastrointestinal cancers, mostly colorectal. These weren't early-stage patients with options. These were people who'd exhausted every treatment available. Their cancers had spread throughout their bodies, despite surgery, radiation, chemo, and multiple rounds of the latest immunotherapies. The process wasn't simple. Doctors surgically removed tumor samples from each patient. Then they isolated the tumor-infiltrating lymphocytes, the T cells already trying to fight the cancer. Using CRISPR, they edited out the CISH gene from millions of these cells. After growing billions of edited cells in the lab, they infused them back into the patients. What happened next surprised even the researchers. Half the patients saw their tumors stop growing. In some cases, the cancer froze in place for 1 or 2 months. For people with aggressive metastatic disease, even slowing progression counts as a win. But one patient's response was extraordinary. She was a young woman with colorectal cancer that had spread throughout her body. Nothing had worked. Within a month of receiving her edited cells, her tumors had shrunk by 50%. By 2 months, they were gone. Completely gone. That was over 2 years ago, and they haven't come back. Let that sink in. Stage 4, metastatic colorectal cancer, the kind that kills 90% of patients within 5 years, vanished after a single treatment. She went from needing constant pain medication to living a normal life. She only needs imaging every other year now to make sure the cancer doesn't return. This wasn't luck. The edited immune cells are still in her body, patrolling for any cancer cells that might try to come back. Unlike chemotherapy or radiation that damages healthy tissue, or immunotherapy drugs that need constant infusions, this is a permanent upgrade to her immune system. The safety profile was remarkably clean, too. All 12 patients experienced side effects, but they came from the standard chemotherapy needed to prepare their bodies for the cell infusion, not from the gene editing itself. No cytokine storms, no neurotoxicity, no runaway autoimmune reactions. The edited cells did their job without causing collateral damage. Now here's where it gets really interesting. This approach is fundamentally different from how we've been treating cancer. Traditional treatments are like renting protection. You need to keep paying with repeated doses. This is like buying permanent bodyguards. One treatment, and your immune system is upgraded for life. The researchers specifically chose to edit mature T cells from the patient's own bodies. This means the genetic changes can't be passed to children. It's a somatic edit, not a germline edit, which sidesteps many ethical concerns about human gene editing. Dr. Emil Liu, the trial's principal investigator, put it simply. Stage 4 colorectal cancer remains largely incurable. This trial brings something genuinely new to the table. His colleague Dr. Brandon Moriarty explained why CISH was the perfect target. It's been holding back our immune systems from their full cancer-fighting potential, but because it works inside cells, we couldn't touch it before CRISPR. The remaining 11 patients weren't as fortunate as patient 22. 10 died from their cancers, though several saw their disease progression slow. One died from sepsis unrelated to the treatment. These were extremely sick people with few options left, so mixed results were expected in a first trial. But even partial responses in such advanced cases suggest this approach could work much better if used earlier in treatment or in combination with other therapies. The University of Minnesota team is already planning expanded trials. There's a catch, though. This treatment is expensive and complex. Each patient's therapy cost hundreds of thousands of dollars and took 3 to 4 months to prepare. The startup company funding the trial actually pulled out because they deemed it too costly and complicated for commercialization. But the researchers aren't giving up. They're working to streamline the process, cutting production time from months to weeks and dramatically reducing costs. Their goal? Make this available for under $100,000 per patient within a few years. Still expensive, but potentially life-saving for people with no other options. This Minnesota trial matters for another reason. It was only the second FDA-approved human trial of CRISPR-modified cells for cancer. We're watching gene editing transition from laboratory curiosity to actual medicine in real time. Other research teams are taking notice. The success with CISH deletion is spurring investigations into editing other immune checkpoints. Some are exploring whether similar approaches could work for ovarian cancer, melanoma, or other solid tumors. A newly formed Minnesota TIL alliance is coordinating efforts. There is also interest in finding drugs that could mimic what CRISPR did here. If scientists can develop a pill that blocks CISH function, it might provide similar benefits without the complexity of cell editing. Though, as the trial showed, there's something powerful about a permanent, one-time fix versus ongoing medication. What makes this particularly exciting is that it worked on gastrointestinal cancers. These cancers typically don't respond well to immunotherapy. The fact that gene-edited immune cells could eliminate metastatic colorectal cancer opens doors for treating other resistant cancer types. The timing of this breakthrough is significant. Just this year, the pioneers of CRISPR technology received the Breakthrough Prize, science's most lucrative award. Their work has moved from theoretical possibility to saving actual lives in under 15 years. That's lightning speed in medical research. For cancer patients and their families, this trial offers something precious: hope. Not false hope based on hype, but real evidence that we can reprogram our own immune systems to win battles they previously lost. One patient's complete remission proves it's possible. The next few years will be critical. Larger trials will test whether these results can be replicated. Researchers will work to identify which patients are most likely to respond. Engineers will streamline the manufacturing process. Regulators will establish frameworks for this new class of living medicines. We're entering an era where cancer treatment isn't just about poisoning tumors or cutting them out. It's about upgrading our natural defenses to do what they've always tried to do: protect us. And this Minnesota trial just showed us what that future might look like. Like and subscribe if you enjoyed the video.